I've been involved in a lot of things over the years, and it's probably one of the reasons why I've kind of got this blend of science and practicality, and the desire to make the great things that happen in research laboratories and try to make it available and practical for people to use who are managing the land. This is my colleague in the business. Maria's also done a lot of different things over the years, and so basically what we do is we help people to grow healthier crops in more efficient ways by better managing soil microbiology. That really sums it up in a nutshell. So what we're talking about is making more efficient use of inputs, whatever they might be. They don't have to be from off-farm, they can be your on-farm inputs, and getting more consistent outcomes. And a key factor of that is making more profit and being able to relax and enjoy your work. So if we could think about the soil, and if you look at soil health, we can think about it being sort of consisting of three things. Here we're talking a lot today about this thing, but it's really all of those things, and all of those things are linked together, and they all need to be working together, and they're all interrelated, and you can't affect one of these things without affecting the other. But what's tended to have happened over the last number of years, come on slide too far, is that most of the knowledge that we've gotten is around this side of things. This is all the nutrients and maybe a bit on the structure, but this area has been largely forgotten about, and probably because until recent years we didn't really have good tools to look at it. But now we've got some better tools to look at it. And I'm going to be sharing with you some of the results, as Rebecca introduced, some of the results from this very group today. And we can have a look at those and start to think, what do they mean, and what can we do about them? Because we have to put those ideas into practice. So what we do basically is we specialise in soil microbiology analysis, also compost and fertilisers and things as well. We try to be focused on outcomes for people. We use advanced techniques. We look at what's being used in research science and try to come up with a practical way to make that available to people. We have quite an extensive range, and we're continually looking at new ways and what new things might be available and how to use them. So if you've got any interest in that, we might be able to help you. Please come and find me afterwards. Okay, so on to the test that the group here did, and I'm just going to run you through some of the tests. The first one that they did was this, what we call microbe activity wise, and it's really a general type of test. So it's basically measuring how active, or the potential activity of the microbes are in the soil by measuring their respiration. So basically, a bit like you and I, how hard they're breathing, and that depends on how much food they've got and the environment that they live in. A bit like us. So you get a good overall picture. It won't tell you what groups there are, but it'll tell you how active they are. And so that's basically measuring how all the sum total of microbes are doing in the soil. Then we've got a test called microbe-wise, and for the purists, we started out like this, and we can measure quite a number of different microbes in the soil, and these are some of the best studied microbe groups in the soil. So we understand quite well what they do in the soil, and there's quite a bit of information about them and what they do and how we can manage them. So we started off with this, and we got all the numbers here, the biomasses of the different groups, and we did that for quite a few years, and people used to say, oh, that's great, that's really interesting, I find that very interesting, but what does it actually mean? And so in order to try to turn that into something more meaningful for people, we created this dashboard on the front here, which are soil indicators. So they're really just indicators. They're not really measuring these things, but they're related to the different biomasses of all the microbe groups that are there and the roles that they play. So they give you some indication, just help to translate those numbers into what that might mean for your soil when you're trying to manage it, which is important. So we've got a number of indicators there, and things like nutrient solubilization and drought resistance and nutrient accessibility and so forth, and then a kind of an overall balance at the bottom. And of course we've got all the numbers when you want to get into it in a more detailed way. The other test that, that the group here requested was one called NWISE, and we've talked a bit about, we've heard an introduction about nitrogen cycling and that type of thing this morning. Well, we've looked at all the scientific literature and all the different ways that you can do this and talk with people, and we've developed a test where we actually measure what's happening at these different stages along the soil nitrogen cycle. So, has anybody seen a diagram like this, the soil nitrogen cycle? Not many people? Okay, well, just to basically, I don't want to kind of intimidate you with information. It does look a little bit busy, but we've thought this is the best way to communicate this information. So basically we've got, starting in the atmosphere, we've got some nitrogen in the atmosphere, and there's a couple of ways that that can get into the soil. So one is by rhizobium on legumes, and the other way is through what are called free-living nitrogen-fixing bacteria. So they can live with, you know, grasses, all types of plants in the root zone, and also fix nitrogen. It can also get into the soil through, well, we've obviously got some in some plants, maybe we've got some manure, and that can also go into the soil as organic matter and be decomposed through decomposer microbes into this organic pool. And then that of course gets converted into mineral nitrogen, which is what's available for the plants to uptake. So there's a process that goes through there, and there's a few different microbe groups, but we can actually measure that rate and see how you're going on that compared to some benchmarks. And so that process can cycle on that way. So just to zoom out a little bit so you can see a little bit more what's going on. It's cut off a little bit on the bottom of the screen there, but I just wanted to show you, and others have made the point this morning, that we've got this organic nitrogen here in this pool. So this can be nitrogen that's fixed by free-living nitrogen fixers, or it can be things like manure, nitrogen in manure and organic matter in this organic nitrogen pool here, and then that gets mineralised or released from that and becomes mineral nitrogen in different forms, which is available for plant uptake. And so the point is we can measure all along those steps, and we can start to get a picture of what's actually happening in the soil. And so we can measure all those different steps. We can measure how much nitrogen is actually being fixed by these free-living nitrogen fixing bacteria, and that's essentially the free nitrogen that you're getting from the soil. And we can measure how much of that organic nitrogen is being mineralised into the plant available N over here. And of course we can measure the total N as well. So we'll get to that in a little bit more detail when we start to look at the actual results. And of course there's quite a number of pages here because this is a little bit more complicated, but basically we're looking at the carbon and nitrogen ratio, total carbon, total N, things like that. And we put all this in kilograms per hectare. And then, of course, all of those things depend on temperature and soil moisture. And so there are a number of good studies around that have been able to relate how much and how fast those things happen at different temperatures and moisture. So you can use that information and you can plug it into this little calculation table, relatively simple, knowing how much nitrogen you need to grow your pasture or your crop, whatever it may be, and you can actually work out then based on how much nitrogen you're getting, and you can plug that into this table, and you can say, right, okay, well, I know I need 200 units of nitrogen. Hey, you know what, I'm actually getting 150 of that out of my natural system, so maybe I don't need to buy all that extra nitrogen that's I'm being told to buy after all. And so that's a way to increase efficiency. And then you get some comments on the back sheet as well. Okay, now on to the actual test results that we've done for the group here. So the first test was the activity test that we talked about. So this is the kind of overall one that measures the respiration or the activity of the microbes overall. And so we've got two samples, and we had sample 1 and sample 2. And I'm not going to reveal any names, but we've got a little bit of background information on them. And so sample 1, we had fertilisers. We had triple superphosphate added at 110 kilograms per hectare per year. That's the history. We had some herbicides added, this type of herbicide combination. And the grazing strategy, these were pastures by the way, the grazing strategy was a set stocking density. In contrast, we had sample 2, which had a different fertiliser treatment. So the main fertilisers applied to this pasture were composted manures and with added gypsum, lime and some trace elements as well. As far as I know, there weren't any herbicides used, and the grazing strategy was termed by Melissa, who sent the tests in, as strategic. So I'm thinking that means rotational grazing and that type of thing. Okay, so just to have a look at them, and we've made this easy because there's a nice shiny dial there to look at. Straight away we can see they're reasonably the same. Perhaps sample 2 is a little bit higher than sample 1, and then that relates to things like how much soil microbial biomass carbon is in there. But generally these are the same. Maybe sample 2 is a little bit higher, and we'll discuss all these results in more context and together as we get along a little bit. So the next test that we did was the microbe-wise test. That's the one with all the indicators and so forth. So just to give you an overview, and of course we've colour-coded these according to our guide, and I have to stress, we always stress that this is just a guide. It's based on quite an extensive survey of whatever literature is available in the scientific published literature and from our own experience, but it really is just a guide. We need to put a peg in the ground. If we don't have a guide, people don't have anywhere to stand. So this is what the ideal soil might look like. And we're currently working on improving that for averages for different industries and different crop types. But for the moment, we've just got this guide. So, and it also makes it easy to compare. So you can see in sample 1, this is the paddock that had the triple super and herbicides, set stocking density. Things are looking pretty good. There's a few little anomalies there, but generally things are looking pretty good. Over this side, things are also looking pretty good. So not a huge amount of difference in the microbe groups that are there. Even the diversity is very similar. And we have to remember that, we're just looking at 2 individual samples here. There's not any replicates, but we'd say those 2 soils are fairly simple. So if we kind of graph those up to make them a little bit easier to compare, we can see that the sample 1 in the green here and sample right, and in here we've got the biomass, an estimated biomass, a calculation on the vertical axis here, and on the horizontal axis we've got the different microbial groups. So just looking at all these that are on a similar scale, we've got the total microbes, total bacteria and total fungi. So you can see actually in sample 1, they're slightly higher. So you might say, oh okay, there's a little bit higher here. But again, just 2 samples, it could just be within the bounds of natural variability. However, when we look at these other, when we drill down a bit and look into the specific microbe groups, we can see a little bit of difference. For a start, in these groups here, we can see that sample 1 having slightly higher, and for some reason on this slide, not all the groups have shown, but let's just say that some of the groups here, this looks like this would be gram-negative, so this is gram-positive bacteria. This would be actinomycetes, slightly higher in soil 1, in sample 1 than in sample 2. But what I want to draw your attention to is a little bit higher. If we kind of look at the next slide, hopefully. Oh, okay. Let's leave that for a little bit later. Looking now at those indicators, those soil indicators that we've got, we can see that both of these soils are pretty much in comparison with both of these things. A little noticeable difference here might be the nutrient accessibility due to mycorrhizas here, but otherwise pretty much the same across all those things. Now looking to the, for some reason one of those slides didn't show up there. We've gone from PC to Mac, so that might be why. What I did want to show you was, and what was going to highlight here on the slide, was this difference here. So this is mycorrhizal fungi. So there's quite a big difference between sample 1, so that was our sample that's fertilised with superphosphate, and sample 2. And a little bit difference here. I think this is the fungi to bacteria ratio is a little bit different between those two. So, but we've got some differences here where sample 1 is higher, but we've got a big difference here between the two in terms of mycorrhizas. All right, so now looking at our N-wise test, so we're looking at nutrient cycling and nutrient availability and free nitrogen fixation. We've got over here sample 1, again on the left, and sample 2 here. So here's our triple super set stocking sample and our composted manure strategic grazing sample. And so we can see a few differences here. Everything is a very similar pattern, but there are a few slight differences. For example, we've got nitrogen mineralisation under ideal conditions. This is how we measure it in the lab, and remember then you've got to use the ready reckoner to get back to your kind of field conditions at the different temperatures that you have. But we've got here, under ideal conditions, about 500 milligrams per kilogram of N per month under those conditions, and here we've got about 550. So we're getting an extra 50 milligrams per kilogram, or roughly units of nitrogen being mineralised, made plant available in this sample here. So that's one of the major differences that are in there. All right, so having a little bit of look at the table that's on the second page, and what I wanted to, and hopefully this is going to work on, yep, okay, great, it worked that time. So what I wanted to show you there was one of the standout things. We've got, you know, very similar results between the two, but one of the standout results here on page 2 is denitrification. Does anybody know what I might mean by denitrification? Anybody heard that term before? Okay, so what it means nitrogen loss. So when you've got anaerobic conditions, you're going to be losing nitrogen out of the system. So you don't want that, obviously. So if we compare, again, these are just 2 individual samples, but if we look at that, in this sample we had some denitrification going on. We had about under these ideal conditions, we had about 3,000 units of nitrogen per month being lost. Now, if you took that back to what might be typical soil conditions, you could probably divide that by about 4. So, you know, probably looking about, well, we're looking about 700 or something rather like that. And then but in sample 2, we've only got about 800 here. So big difference between these 2 samples, and hopefully we'll get to uncover that a little bit as we go on. Aha, this is what might be, this is what I want to say, and the little green yellow circle hasn't turned up in the right place. But yeah, I wanted to highlight here the difference in mycorrhizal fungi and try to relate all those results together. So remember, we had both of the fields had very similar results except for mycorrhizal fungi. So what might mycorrhizal fungi be doing in the soil? And we've already heard from Tim this morning explain a little bit about them. Here's a diagram of a soil aggregate, and hopefully you can see that up the back there as well. And basically these green things that you can see through here are the mycorrhizal fungi, and they're binding all the other sorts of aggregates, plant debris and organic matter, the little brown compounds, silt aggregates, the ones with yellow circles, and clay, etc., and binding those all together. You've got a plant root here, binding that all together and forming a soil aggregate, and when we get a lot of aggregates together, we get soil structure. So what is soil structure? Well, a well-structured soil, if we look at that, we've got lots of big pore spaces in here. So we've got lots of nice big aggregates. We've got lots of spaces, and we've got lots of spaces for air and water to get in all of these parts here, and you know, store water, let the water drain away, and also lots of room for air. But in this poorly structured soil here, where we've got very little structure, very small sort of crumbs together, we don't have a lot of space at all, and what would be, you know, quite rapidly filled up with water. So what does that mean? So when we've got mycorrhizas, and mycorrhizas are a key organism for building soil structure, and they exude this substance called glomalin, which is kind of like a glue that aggregates soil particles together. And often people say, well, what's the most important soil microbe? Well, they've all got their role to play, and we need a diversity there, definitely. But if somebody was asking me what's the single most important soil microbe, if you had to pick one, I would say mycorrhizal fungi, because they do so many things. They form soil aggregates which provide homes for other microbes. They transport nutrients from the soil into the plant. They can bring moisture into the root zone from other places as well. So what might we do to build those up if they're low? For example, in sample 1, they were relatively lower compared to sample 2. So what might we do about that? Well, we need to stimulate them and work with what's there. And if they're not there, we might consider inoculating them. So one thing that stimulates mycorrhizal fungi is soil carbon. So if we can increase soil carbon and we can add soil carbon, we can build up those microbes. So, and other things that we can do is avoid over-tillage. Tilling the soil breaks up the networks of these mycorrhizas and they don't function properly. And avoid over-fertilisation. Too much soluble phosphorus depresses these guys and and stops them from doing their job and colonising the plants, and also particularly systemic fungicides as well. And if all else fails, if they're really missing, you could inoculate with a kind of good quality verified inocular. One thing I forgot to mention before is that we had true anaerobes were measured in both of these soils. So it's not surprising really, because we've had such a lot of rain, and true anaerobes live in anaerobic conditions where there's no air. So we've had a lot of rain over the last season, so it's not surprising that we would have seen some in there. But we really don't want those there, because for a start, they're involved in denitrification, which we don't want, and also they can produce phytotoxic, plant toxic compounds which damage roots and let root disease in. So what might we do about that? Both of the soils had those, and we have had a lot of rain. But if we were going to do something about that, we'd want to improve drainage and decompact the soil in some ways. We've already had some presentations about opening up the soil and so forth. So that could be, through infrastructure, drainage, controlled traffic, changing the calcium-magnesium ratio in the soil if that's an issue, and maybe some mechanical and chemical means. The last set of tests that we got for the group here were kind of conventional nutrient tests. And what I wanted to just draw your attention, they're largely very similar, maybe a little bit higher aluminium in sample 1. But what I wanted to draw your attention to was in sample 1 we have quite a relatively high level of plant available phosphorus, maybe a little bit too much in that system according to the guide here, whereas in sample 2 that's a lot lower. And so what I also wanted to draw your attention to was what's called the phosphorus buffering index, which is kind of a chemical snapshot, and really that should be less than about 100. So both of these are kind of over the guide. So what that means is that this is both of these soils are highly P adsorbing. They're locking up soil, and that could be due in part, maybe part due to the aluminium, but probably more due to the really high iron that's in there. And iron will bind with phosphorus in the soil and form iron phosphate, which is insoluble. Okay, so a summary of results, because we've only got a few minutes left. So in our sample 1, where we've got our sort of conventional fertilisation and strategy, actually most of the results were similar between that and sample 2, which had more manures and strategic grazing. But for example, compared between the two, we had slightly lower microbial activity in sample 1, slightly higher bacteria in the soil, substantially lower mycorrhizal fungi compared to the two. Denitrification was substantially higher between the two. We had higher plant available P in those, and we also had a higher grass tetany risk, which I don't think showed up on the bottom of the screen due to our transfer to MAC. So what would our conclusions be? Well, looking at these things, and again between our sample 1 and sample 2, I'll sort of do the comparison with sample 2 this time, not to be stuck on one, favour one over the other. So in sample 2, we've got comparatively higher total organic carbon and lower plant available phosphorus, and that may be benefiting the mycorrhizal fungi. The higher mycorrhizal fungi may be resulting in more soil structure, and that improved soil structure may be resulting in less denitrification. Remember, there was less denitrification in this soil, and so we're losing less. If you want to convert nitrogen to dollars, you'll know what a unit of nitrogen is worth. You can do the calculation. That's going to all add up. And the higher soil calcium and magnesium in this soil may decrease the grass tetany risk. I apologise, that didn't show up on the bottom of the screen. So, what to do? I was going to use this the time left to ask for some suggestions from the audience. How are we going on time? Have we got a couple of minutes to ask people? Okay. All right. Question time starting now. So, given our whirlwind tour through the soil microbiology, has anybody got, in seeing those results and conclusions, what would you do? Any suggestions? Okay, Tom.
Manage the animals properly. All I do is make sure I manage those animals properly.
That's Tom's suggestion. Thank you, Tom. Any other suggestions? Stunned silence. All right. So, well, luckily, I did my job and I prepared a few of my own conclusions here, and you'll have to forgive me because I'm not a farmer these days, and I never was a grazier, so I thank Tom for his contribution to keep managing the animals. So what are we going to do? If we were going to make any contribution there, one thing might be to manage the soluble phosphorus fertiliser additions, and you could do that using our P-wise test, which basically is the same as the N-wise test, only for phosphorus. But managing that, that might, knowing that suppresses mycorrhizas, that might increase your mycorrhizal, help to build your mycorrhizas up that are naturally there, increase that level, better soil structure, increased drainage, less denitrification. So that's what the strategy would be there. That's just my suggested strategy. We might want to increase soil carbon because that would stimulate those mycorrhizas, which could be increased further, but we want to manage the carbon to nitrogen ratio. We don't want to get in a situation where our carbon to nitrogen ratio gets too high and you end up locking up nutrients. We might want to improve drainage. We had those anaerobic bacteria were elevated in both the samples, and it has been very wet. Sometimes there's just nothing you can do about it, but if it was possible, we could improve the drainage by managing any compaction, for example, if there's any compaction there, or perhaps some earthworks to improve drainage. And for the grass tetany issue, which unfortunately couldn't see, was cut off down here. We might want to apply some dolomite, which would be calcium and magnesium, to bring those levels up in the soil. All right, well, I think that's all from my presentation. Any questions?
Thank you, Ash. I had a question. Do you mean to do that on both soils or the sample, the chemically treated soil? Or do that on both samples? So your suggestions there? Yeah,
I think both soils could benefit on it, but probably the chemically treated soil was the one that had less mycorrhizal fungi and more denitrification. So that strategy would probably have more effect on that farm.
Okay.
Yeah, good point, and well picked up. Yeah, our guide level is the same. As I said, we based it on what the ideal soil would look like, irrespective of whether it's sand or loam or whatever. And that's a big call, but the truth is that there's simply not the information around, for all this stuff to actually come up with guide levels for individual soil types and so forth. It will probably be a life's work. There's so many different factors that can influence. But we're working on it. So the next step would be to have some guide levels for, say, pasture in, southeastern South Australia, for example. And the initial idea is to have an industry average. We've been collecting this data now for about 7 or 8 years, so we've got a reasonable amount of data. And so the next step is to start saying, okay, well, what's the industry average for your area? And that might be a better comparison. So good point, and yeah, we're working on it.